Good morning, everybody. I'm Mike Rice, Head of Advanced Therapeutics at Lumanity. I'd like to thank the organizers for inviting me back to moderate the 8th Annual IO Investor Forum. In this session, we'll be discussing opportunities and challenges for upcoming paradigm-shifting programs that could widen the accessibility to a broader set of patients with heme and solid malignancies. I'm joined by an excellent panel of investors and innovators. So we have Gábor Gyülvészi, Senior Associate at Nextech Investments, Deborah Barton, CMO of Carisma Therapeutics, Paul Lammers, President and CMO of Triumvira Immunologics, and Shelly, Vice President, Business Development and Alliances at Prescient Therapeutics. So I'll let the panelists introduce themselves in a couple of minutes, but I just wanted to set a little bit of context for the discussion today. And I hope that we get into an interactive discussion to really talk about the issues that, that these novel disruptive technologies are potentially addressing to broaden the potential to a broader set of cancer patients. So cell therapy now represents about 35% of the IO pipeline. It's amazing how far we've come and the change in tide from conventional therapeutics to cell therapies. And this doesn't even include the vaccine programs that are out there. These are just adoptive cellular therapy programs and various different iterations and formats. It's already yielded 6 approved autologous cell therapies for B-cell malignancies, as you know, and multiple myeloma. All targeting CD19 and BCMA. And these programs has really set the benchmark for what can be achieved clinically. Now, I know there's a lot of hurdles on the commercial side and making them broadly accessible, but from a clinical benchmark, these are what, are being set for the potential for cell therapies, rightly or wrongly. They have a lot of background and development that led them to these potential clinical benefits. However, these are still accessible to very few patients. We hear things like there's 50 patients waiting for 1 slot of a Abecma treatment at some of the tertiary care centers that are licensed. So we have a long way to go to really impacting the broader cancer markets. So let's talk a little bit about financing. What is driving the innovation in the space? So in the last 2 years, we've seen about 165 financings, $22 billion from seed to IPO in early stage, largely pre-POC programs. $6 billion of this went into allo programs that address scalability and may open up a larger set of solid tumors, dealing with all sorts of functional improvements needed to really make a dent in that space. However, many of these programs are de-risking their platforms, going after validated targets. We're tracking 135 CD19 programs, 28 BCMA programs, and 14 HER2 programs are already in the clinic. So these are very, competitive spaces where proof of concepts being established. Early investors are seeking the quickest path to get into the clinic so they can prove and add value to the programs. However, the question is, where do we go from there, right? Are they just proof of concept opportunities, or is there a broader commercial opportunity for those markets? So the investments include novel paradigm disrupting platforms such as novel cell types, so we'll hear about that today, functional improvements for effector cell and function, host evasion, particularly for the allo stealth programs to make them more persistent and long-lived, TMA modulation, way to deal with the solid tumor microenvironment, tumor targeting, including adaptive targeting approaches, which we'll also hear about today, preventing off-tumor toxicity, preventing antigen escape, and addressing a whole host of resistance mechanisms and relapse. So today's panel will touch on all these aspects. And so, without further ado, I'd like to, invite the panelists to introduce themselves and sort of really get into an interactive discussion about, the opportunities and issues that we just touched on. And that said, before I introduce everybody, in the last, quarter, 2 quarters, we have seen a very different change in access to capital that are really driving some reconsiderations about, how do you move these programs forward and, get access to capital investments and everything. So we'll get into that as well. So why don't we start, and I'll introduce the panelists. Gábor, why don't you introduce yourself and then I'll introduce the biotechs.
Sure. No, thank you. It's a pleasure to be here today. Thank you for the invitation. My name is Gabriel Durbiss, and as you mentioned, I'm a senior associate at Nextech Invest. Just briefly touching my background, immunologist by training, worked on T-cell cytokine biology for a couple of years, joined industry, Roche namely, spent a couple of years there and really worked on different flavors of IO programs, small and large molecule-based ones, and currently working at Nextech and evaluating exciting opportunities, either in the cell therapy space, targeted therapy space, and anything which is connected to oncology. Maybe just a couple of sentences on Nextech. We are a small venture capital firm purely focusing on oncology. We are modality and stage agnostic. We love outstanding management team when it's combined with strong scientific foundation and like-minded investors. This is our simple investment thesis. And just really to point out one more important fact with Nextech, we work with a team of world-class scientists in the field of oncology who really help us evaluate opportunities. And just to name a couple of individuals here, like Bill Kaelin and Charles Sawyers, in the IO space, Phil Greenberg, Tony Rebus, and others. And we really, together with this team, we hope to make the best investment decision and identify the best company, which hopefully can provide a significant benefit to patients and can deliver a drug to patients in need. That's my short background.
Great, thank you. So next I'd like to introduce Debora to give self-introduction, tell us about Carisma.
Thank you, Mike. I'm Deborah Barton. I'm a medical oncologist and I'm the Chief Medical Officer of Carisma. Before Carisma, I did 10 years of big pharma. Then I made the jump to biotech. I worked with radiopharmaceuticals. I worked with Iovance, and now I'm at Carisma developing CAR macrophages. So Carisma is a clinical stage, Philadelphia-based company developing a first-in-class CAR macrophage, treating patients with HER2 overexpressing solid tumors. It's the first ever CAR macrophage in the clinic, so it's really exciting. That's why we went to HER2 to de-risk the target, to really make sure we understand the biology of CAR-Ms in humans. And importantly, we have some data coming up at ASCO. We'll have safety, trafficking, TME modulation, and preliminary efficacy to be discussed at ASCO.
Great, thank you, Deborah. So Paul Lammers, why don't you introduce yourself and tell us about Triumvira?
Okay, thanks, Mike. Paul Lammers, President and CEO of Triumvira. I'm a physician and biologist by training. I've been in pharma and biotech for over 33 years as a drug developer, brought several products to the market. First part of my career, I was chief medical officer of different companies. Then I came to Austin, Texas, and became president and CEO of Therapeutics, a microRNA company. And from there on, I became president and CEO in 2018 of Triumvira Immunologics, an exciting company based on a basically new and very well differentiated technology known as TAC, or T-cell antigen coupler. We are in the clinic also targeting HER2, just like Deborah and Dan will talk about. And I think that makes perfect sense. We'll get to that later, Mike, no doubt. But yeah, it's an exciting, we made a lot of progress when I joined. There were 2 scientists in Jonathan Bramson's lab up in McMaster University who are employees. Right now, we have 56 people. We'll be about 90 people by the end of the year, which is really exciting. So we're building a broad pipeline focus solely on solid tumor targets. And we are using both an autologous as well as allogeneic approach because we think that both platforms are absolutely critical for building a strong and broad cell therapy company.
Great, thanks, Paul. So Dan, why don't you introduce yourself and tell us about Prescient?
Sure. Thanks, Mike. Pleased to be among all of you today. Dan Shelly, VP of Business Development Alliances for Prescient Therapeutics. I am a PhD, MBA, molecular physiologist, actually, but over the last several years moved into advanced cell and gene therapies. And now as part of Prescient, Prescient is a small Australian-based but publicly traded company. We do have a portfolio of some small molecule targeted therapies, but we're here because of our OmniCAR platform, which is sort of an intentionally, rationally designed platform that is a universal immune receptor platform. It's the only covalent system of its kind in development. And really what we're focused on is switching the paradigm, if you will, from pre-selecting an antigen target because you think it might be the best opportunity to actually having modular control and allowing, if you will, the patient's cancer to dictate what treatment you provide. So we have the ability to create post-infusion control of these immune cells, and we can redirect them or multi-arm them against various antigen targets. So we think the biggest issue right now in this field is really antigen escape and relapse. These patients don't have many options after that, and we think that OmniCAR can address it.
Great, thanks, Dan. All right, so why don't we get into the discussion? And I'd like to first start off with the investor perspective. So why are investors, so interested in this next generation, assuming that things are going to continue going forward in 2022? But why have investors been so enamored with these novel technologies and investing at a very early stage in these programs?
And maybe just stepping back a little bit, I think first generation T-cell therapies, they prove themselves to be extremely efficacious early on. And in certain patients, durability is excellent. But unfortunately, as you know, not all patients respond to those treatments. And what really makes us exciting is really the potential to provide to those remaining patients potential cure with second and later generation T-cell therapies. And as well, I would compare somehow T-cell therapy or the cell therapy space to the antibody space in its infancy 20, 25 years ago, when there was the first product on the market that was a murine antibody. It did its work, but it didn't become commercially successful and it had liabilities. And I think that is somehow similar to first generation T-cell therapies. The liabilities, it's not yet a commercial success, but it's slowly advancing towards a commercial success. And now we understand what liabilities those products have. So I think with second generation T-cell therapies and cell therapies in general, you can kind of address those liabilities, you can fix those liabilities. And this makes us exciting really that we can select from an amazing pool and pick the best suited approach, because there are so many ways to fix, you can deliver the CAR differently, or you can deliver not a CAR, but a TCR or a different type of receptor. You can arm those cells. You can pick outside of the T-cell pool as well a different type of cell. And there are so many things to explore that we are convinced that sooner or later one or probably more than one companies will fix those problems and really create a commercially successful product. And this is just maybe on the science, but as well we know that manufacturing is a big hurdle for T-cell therapies to become commonly available. So we really pay attention to this angle as well, how those products can be manufactured, what are the steps which can be implemented to shorten the manufacturing time, and how you can make that more economical in a way as well. Because as an investor, when you look at the investment, you invest a big part of your money into manufacturing in each and every company. And that's still a liability from our end, because we have to commit a huge amount of cash for manufacturing. And we would love to rather provide that to scientists and to science to really come up with great ideas to advance those therapies further. However, at the same time, actually, because there are so many ways to engineer those cell products, that we are kind of like struggling a little bit with this landscape. How do you kind of really follow those companies? Because there are so many. You mentioned how many clinical trials are there in the CD19 and the BCMA and so on with those targets. And we just have hard time picking the winner one, because we just don't know which method will become successful. We very often feel that there are small incremental differences between companies, and we are really trying to identify companies which make a groundbreaking kind of discovery, or like a bigger difference, not just an incremental, but really a significant difference. But that is extremely challenging in this current environment.
Right.
So it remains extremely exciting for us, the cell therapy space, but because of this variety of so many different flavors, makes us extremely challenging the investment and find the most suitable company for our investment, for our portfolio.
Right. Great. I think you touched on many of the points. So you highlighted, the opportunity, right, that we're just in the sort of early generation that, of seeing what could be fulfilled if we can make it more broadly accessible. You pointed on many of the liabilities that are, challenging the field right now, and highlighted many solutions, right? So I guess, the question I think, is before we turn to the biotechs is, I think we want to understand, you talked about this very complicated landscape, and it's hard to understand, what are the real viable products that are really going to lead to a real viable solution to any of these challenges. What sort of questions would you like? What do you want to hear from the panelists today that could give you more confidence in the opportunities that they present?
For us, really, a major question is the durability of the response of different cell types other than T cells. We understand T cells reasonably well. We know that they form memory. And there are a couple of papers from excellent investigators showing that those CAR T cells persist in the body for a decade even, and can be reactivated and can trigger an immune response against reemerging tumor cells. But our struggle is often to understand like NK cells, macrophages, gamma delta, other cell types which don't necessarily form memory, at least not in a way as we understand memory, how long those responses will last. And can you really take advantage of this more than one administration, this sequential administration of the product, and really provide it as kind of as a small molecule in a way, whenever it's needed, the patient comes in and it takes the medication or the treatment. The durability is a big question mark for us. The second is we often struggle understanding, like when certain companies go after certain mutated neoantigens or oncogenic drivers, how fast resistance will emerge, and what can you do to address those resistance mechanisms? Because we have no doubt that resistance will emerge. We see that through different mechanisms. And I would love to understand better your view there, like what can you do to, again, provide a longer lasting benefit to those patients who develop resistance? And maybe the third, which is important for us, is manufacturing, and kind of how can you streamline manufacturing? Do you think there will be a time when the patient comes in and can immediately get the drug or the cell therapy product, or you always have to really harvest T cells because that will be the best way forward, or any other type of T cell from the patient? Or off-the-shelf therapies will be available and will become successful as well. We see some setbacks in the off-the-shelf therapy space as well. And I would just like to understand your view on those setbacks and what's the way forward. Mm-hmm. These are, I think, kind of a couple of overarching questions and doubts on our end.
Great. Excellent. I want to come back to the situation with the capital markets in a little bit, but I think I'd like to turn over to the biotechs really to react to some of those points. And we have an interesting sort of case study here because I didn't really plan the panel this way, but I think everybody has a Her2 program for solid tumors, which makes this kind of an interesting, real world example of it. And I believe both two programs are in the clinic already. I don't think Dan, Shelly, I don't think Prescient's in the clinic yet. But maybe we could just start off with the clinical stage programs. And Deborah, maybe you could talk a little bit about your goals with your Her2 program. And I'd love, Paul also to add to that.
I would love to answer some of Gábor's questions. So thank you for that. I think it's important to understand the macrophage biology and the capacity of the macrophage to recognize the target. And in this case, it's Her2, but it's just a Trojan horse because it will identify the cell, phagocytose, and kill that tumor cell. And macrophages are professional antigen presenting cells, so they will be able to present all antigens that are extracellular and intracellular to T cells who will then mount a long-lasting immune response. So that's where it's not a one-person job, it's a teamwork. So that's where we believe we're gonna have long responses. And also on the point of resistance, because we know tumor are heterogeneous, and if you hit on one target only, the other clones will emerge. And because the macrophages have this interaction with the T cell and process and presenting all different antigens that can make a difference into antigen negative escape. So those are points that are, I think, very important, going back to the biology, not really focusing on the target itself, but on how the cell works. And just a quick point on manufacturing, we're working with in vivo with the Moderna collaboration, and also looking at allos. So we do recognize that different manufacturing procedures will be important.
Mm-hmm. Oh, can you tell us a little bit about your Her2 program too?
Yeah, sure. So look, there are several reasons why picking Her2 makes a lot of sense. First off, if you would take an unvalidated technology and you take a new and unvalidated target, then the data are very difficult to interpret. The oncologists are very happy that we picked Her2. Why? There's still a high unmet need. We know that Her2 has done fantastic and is moving up the chain. We know that there's about 11 products approved now for breast and 2 for gastric. However, there's, if you look in the US, about 50,000 patients die each year of HER2 overexpressing tumor. That is not just breast and gastric, that is endometrium and gallbladder and lung and pancreatic and go down the list, right? So, and they don't have any approved HER2 targeted therapies yet. So there is a high unmet need. Oncologists are happy and they like the fact that the cell therapy is a late stage single potentially curative treatment that is really appealing to them and appealing to patients, as you can imagine, who've already been to hell and back, so to say, with different therapies. So the other thing is just to mention, it addresses a bit Gabriel's question about or concern about the antigen escape. The nice thing with the cell therapy, you're not affecting the intracellular HER2 signaling pathway. Whilst we all know that tumors have a fantastic way of mutating around the block that you put in place in a pathway, we just need some HER2 to stick out the cell for the cell, T cell to latch onto and kill the cell. And we have done those experiments where we take cells that are not HER2 overexpressing, we're adding increasing amounts of HER2, and even at very low HER2 expression, we see that the cells are killed. So I think that is another nice way of a cell therapy approach that does not affect the signaling pathway, so it minimizes the chance of an antigen escape in this respect. So for us, the Her2 study is a perfect clinical proof of concept. It's a validation of the technology platform. The nice thing also for us is we all know that the last few years, because of the pandemic, it was very difficult for any oncology company to enroll patients because of ICUs were all taken up by COVID-19 patients. Sites were shut down, and clinical research staff was sent away, and they had to rehire and the whole thing. But this year, recruitment has gone ballistic almost, which is fantastic. And it shows that the sites, we have MD Anderson, Dana-Farber, et cetera. So they're all extremely excited. They say, look, there's very, very few exciting cell therapy studies in solid tumors. And so they're very happy to participate. So I think HER2 for us made a lot of sense. And obviously now it's all about the data, right? I mean, and it gets back to the investment thesis as well. Last year, a lot of we did many investor meetings, and they all said, look, Paul, we love the technology, we love the team. You're starting your study, so let's just wait for data, right? I mean, that's the next best thing, if it's not the best thing. So anyway, so we're in the process of assembling that. So we'll have, we'll have data in the middle of the year. We hope to have a recommended phase 2 dose by the end of the year. It goes into dose expansion. And the other thing is we're adding a combination arm to the study as well, by Pembro or Nivo, depending on our discussions with Merck and BMS, because we also know that oncology is the world of combination therapy as it is. And, the question always is, can you further enhance persistence, if you add, an anti-PD-1 or PD-L1? So, so I think that we're very happy with our HER2 program. It's a great lead program. And also, look, there's so much other data out there. You can compare your data easily with other modalities. And I think that is a big plus as well. Look, we're excited about our other targets like Claudin 18.2, Gucci2C, GPC3 in the pipeline, but there's very little data out there, clinical data out there. So it's difficult to interpret.
Right.
That's not the case with HER2.
Great. Dan, why don't you tell us about your approach and, some of the, how you're addressing some of the questions that Gábor.
Yeah. No, I appreciate that. Those are actually really important questions. I do think that Prescient's approach with HER2 is very similar. It's more or less a proof of concept. It allows you to compare against known data. I think for you to understand Prescient's approach, I just have to back up a second and describe how we design the system. We don't actually make a CAR immune cell in the first instance. We express, instead of a CAR, a basically untargeted receptor. We separately or simultaneously then administer a targeting ligand that has the other piece of the covalent system. So we decouple the CAR from the cell. Now, this has a number of advantages that directly address your questions. In terms of durability, most of these immune cells are constitutively active all the time, right? They're activated and they're on, period. With the OmniCAR system, we can turn them on, we can turn them back off, we can turn them on again. So we can stimulate them metronomically, sort of the way they're naturally acting in the body. So we believe that this approach will enhance durability of cells because they're not exhausting. In addition, with our system, when you activate these cells and they replicate, they proliferate, they don't make new CARs, they make new unarmed immune cells, which can be reactivated later. So you're basically effectively replicating the therapy in vivo every time you activate it. All right, so that's one advantage of durability. And we think that applies, this system applies to any cell type. So the other piece of it is there's whatever intrinsic engineering either Prescient or our partners would impart on the cells themselves, plus this sort of metronomic on-demand stimulation we believe is going to improve durability. In terms of new targets, yes, we have the HER2 program. And again, it's more of a proof of concept, but the way we look at targeting, because you can turn on, off, and back on again, you can redirect, which means that we can create an immune cell, and if we clear out 80% of the cancer cells that express, say, HER2, and now the cancer is expressing a new receptor, we can simply administer a different targeting ligand and redirect the cells that are already there. So we don't actually have to re-administer the cells each time we want to redirect them. Or we can multi-arm these cells with multiple targeting ligands for highly heterogeneous cancers. And I guess the final piece you mentioned about manufacturing, with a conventional approach, for each target you want to go after, you have to make a new de novo CAR cell, right? Which means if you had 6 targets, you'd have to have 6 manufacturing runs, 6 INDs, 6 release criteria. With the OmniCAR system, you make 1 immune cell. You have one IND, one optimized manufacturing process, and then you have a library of targeting ligands against the most prevalent antigen targets in that cancer. So that allows us to address sort of the most prevalent antigen targets, clear those out. You know, these patients are in the clinic, they're being evaluated constantly. You're taking tumor biopsies just to see progression, right, and clearance. And you can determine, oh, well, now they're expressing, the cells that are left are expressing this antigen target. I can redirect these cells from one target to another, and I can cure these patients. So we think that this approach is significantly enhanced for manufacturing because it allows for optimization, it allows for off the shelf. We think it has regulatory advantages. So that's how I think we would address those questions that Gábor provided.
Great perspectives from all of you. So I'm hearing, you know, HER2 was chosen because it's a very validated target. You can compare to other modalities, and it's proof of platform. So you're adding value to the platform. You know, in itself may be a commercial opportunity with, I think Paul mentioned 50,000 HER2 positive advanced solid tumor deaths. You know, even as this market grows into HER2 low and low target density, you know, that there may be, you know, even other segments of the market that could be addressed. So one, I just like to hear your perspective in, you know, with all these HER2 cell therapies now coming in, you know, and all your different approaches, you know, how do you educate investors on your differentiation story, you know, within HER2 positive tumors, and then we can sort of get into your next iterations. Anybody want to jump in there?
Well, I'll let Deborah go first.
Thank you. I was opening my mouth. Yeah. Well, I think it goes back to 2 points. One is the cell biology that we're using, and each one has something different. And the other one is the tumor biology and how that particular cell can attack those aggressive and heterogeneous tumors. So I think that's the real differentiator. And in the end of the day, of course, it's going to be safety profile, efficacy profile, and the feasibility of bringing it up. So it's really a package, right? Everything is important. So yeah, keeping in mind the tumors, they change, they evolve over time. So your platform needs to be able to address that. And of course, I believe macrophages are uniquely prepared to do that because of the way they attack the tumor cell.
Mm-hmm.
Yeah, for me, it's all about data talks, right? Clinical data talks. So obviously, you know, once we have, right now, we are enrolling patients that are 1+, 2+, or 3+ HER2 overexpressing. And as I said, we're adding also a combination arm to the first phase. Once we have a recommended phase 2 dose, we're going to dose expansions based on the results that you see in your, you know, escalation, right? So, and as I mentioned, the 50,000 deaths, Mike, I mean, 36,000 of those are non-breast or non-gastric patients, right? So basically on the results that you see and the indication, you can get it pretty quick to an approval and say, let's take an example. If you have a HER2 overexpressing pancreatic cancer group and you see results there, I think you can have a really constructive discussion with the FDA about what kind of program you need to do to show that, right? You don't need to have the old fashioned, you know, thousands of patients in oncology phase 3 trials to get to an approval. So I think that makes a huge difference because FDA is very interested in new targeted therapies, you know, that are for specific tumor types. And I think that's really the best road to go.
And for, you know, for Prescient, what's interesting, while we do have the Her2 program, it's very likely that our AML program, where we have 2 and potentially a third antigen targeting ligand, will be actually our first to clinic, which we anticipate early next year. Part of that is because we really want to show the utility of this approach in a highly heterogeneous cancer. And, you know, in AML, there's at least 28 different antigen targets being investigated in a CAR T format or CAR format right now, which really clearly shows you that this suggests that no single target, maybe not even any dual target, will be sufficient to treat that cancer, right, or at least get all of the patients that you are treating, right? And so I think that's where we're going to focus our clinical efforts. The Her2 program will continue, and we'll include that with other antigen targets like EGFR V3, where we have a program in GBM.
Mm-hmm.
Yeah.
Great. So,
Mike, if I can talk still a bit about manufacturing, because as Gabriel rightly said, that is still a huge issue in this space, right? Whether it's for investors or for companies or for boards, So I think, and the last few years, whenever we met with VCs, they've always said it has to be allo, and allo. And we all understand the benefits of allo because you have a shorter time to therapy. You're taking cells from healthy donors, right? You create massive cell banks, all of those. But Selectus, allogene, and as Gabriel himself said, there are some issues with allo, right? Even now, the key opinion leaders in CD19 said, look, I get 1 month efficacy out of allo CD19. I get 5 months with Kymriah Yescarta. So guess what I'm picking, right? So, but that's important. So we think that having both autologous and allogeneic platforms is really critical. And it gets back to who ultimately is our target population. More than 50% of all cancer patients in the US are treated at community oncology centers, not at the highly, specialized cell therapy tertiary academic centers, right? And even Gilead as well as Novartis admit that their sales are below expectations. Yeah, look, we would take as companies $200 million a quarter every day of the week, right? But it is not the billion-dollar products yet. Why? Because of the limitation on the number of patients that they can reach, right? You mentioned also Abecma, right? 1 out of 50. I mean, so we need to get to a point, and that's why we're very happy that we're using Lonza's Cocoon system, because it's automated, closed. The yield and the quality of our tech T cells is fantastic, way higher than with the standard industry back method. And ultimately, this machine was designed, it's not there yet, where we can put it into the community oncology center. Patient comes in, gives us blood, comes back 3 weeks later, gets the cells back. What would be better than that? That would really expand the world, whether it's for heme or for solid tumor, type. So I think that is really, really important. And now investors are very happy that of course, we did the deal with Lonza. We're the first biotech in the US to dose patients with cells made in a cocoon, and it's been great. So now people really like the fact that we both have an autologous and an allogeneic platform. Right. So I think that, at the end of the day, look, we're developing gamma delta cells as our allo platform, which is also very intriguing. And we've shown preclinical proof of concept. So we'll bring that into the clinic as well. But the durability, with these gamma delta cells is still uncertain compared to what happens with standard alpha beta cells. So I think it's important for us that we develop both and have, perhaps even multiple INDs on the same target, but with an allo and an auto approach.
Yeah. And it's a good point, many of the points that you made there, but, I think most of you have the luxury of not being on the public markets, but those initial allo programs, both, alpha beta T cells and NK cells, the gamma deltas have fared a little bit better, but, they've all lost like 80-90% of their market cap. So, and that's with, proving what they want to prove with proof of platform. So the question is, and I'd like to turn it back to Gábor for a second, is, what I'm hearing is that, so there's a lot of segments that go within the Her2 market, right? So there are commercial positionings, even though it's going to be very competitive with all these programs going there. But the real value is in the next iterations of the platform and the products, and even with combination therapies with PD-1 and other things. So the question is, private investors, want to see a timeline as quick as you can get it to IND and into the clinic, right? Whereas the public investors, like you said, Paul, you're being judged on the data, right? The clinical data is speaking. That's all they care about. So, Gábor, so how are investors, seeing this? Are in this today's environment? Are you patient enough for iteration to your next products?
So, just speaking for Nextech, we are patient enough. And actually, we call it currently, this is the biotech nuclear winter, what we are facing at the moment on the public markets. And when it comes down to financing as well, and availability of capital, this is a hard time now for biotech companies. But I think with patient investors, many companies will survive who can show clear differentiation in the clinic and clear efficacy signal in the clinic. So that's what was mentioned earlier. For us on the public side is really the clinical data what matters. And this is what we want to look at. And this is how we can judge companies. Because unfortunately, preclinical data and a given number of portfolio programs, INDs, first patient-ins, those are nice signs of progression, but ultimately you have progressed, but ultimately you have to show clinical efficacy data to convince investors and maintain valuation and be able to raise additional capital to continue advancing your programs and hopefully provide really benefit to patients.
Is this going to drive more risk-taking, I guess, more skipping steps in iteration, taking on more, risk to get to a more differentiated product, and more careful, indication selection,
I'm not sure whether we are, as an investor, taking more risk. We are definitely taking more time to evaluate opportunities.
Yeah.
And maybe in that case, we are taking even less risk, or we try to really find companies which have proven already their clinical hypothesis. So they de-risk the asset in a way, and we create a position in those companies. If you just look at the number of companies which are out there on the public markets, there are around 900 biotech companies. 40% of them, they have cash runway for another year and a half. So it will be interesting to see what happens in the next 1.5 years. I believe we will see many mergers and many companies cease existing in the near future because they just can't raise enough capital because they don't have clinical data to prove their point and convince investors. So taking everything into kind of consideration, we are taking more time to evaluate opportunities, and we became a little bit risk averse and valuation sensitive. And I think many other VC firms think similarly.
Mm-hmm. Great. Anybody want to provide their perspective?
Well, Mike, I know that, we've been, reminded the fact that, we've already gone over the time. So I'm sure if the recording is still going on, but, we can talk for hours about this stuff, guys.
Yeah.
It's great. Yeah. And I really appreciate Gabor's perspective, and of course, Deborah and Dan as well. So
great. All right. Well, I think we're out of time. So if anybody doesn't have a last remark, then I think we can conclude today's panel.
Awesome. Great.
Thank you, everyone.
Good to see you guys.
Thank you as well.
Bye-bye.
The chat.